PCR is a magic wand that molecular biologists wave over DNA and amplify it. So hello and welcome. I'm your host, Dr. Ruby Gill, and I feel privileged to extend my warm welcome to all on the behalf of lovely professional university. So attendees, you might be wondering the reason of gathering virtually. So let me reveal the reason. We are here to witness an incredible session on the topic PCR, a photocopy machine for DNA. I'm sure you're as excited as I am for today's learning process. But before that, allow me to introduce the experts of the day. Dr. Neeta Raj Sharma, an intellectual personality, a leader par excellence, and professor and dean of School of Bioengineering and Biosciences, lovely professional university. Ma'am, it's a privilege to have you on board with us today. Thank you so much for joining us. With equal pleasure, I would like to welcome Dr. Umesh Gautam, Professor and COD, Molecular Biology and Genetic Engineering, School of Bioengineering and Biosciences, lovely professional university. And furthermore, we have with us Dr. Gurmeen Rekhra, Assistant Professor, School of Bioengineering and Biosciences, lovely professional university. So thank you, ma'am. Thank you, sir, for your valuable time. Additionally, we have with us our admission nominee, Ms. Rachna Bajaj, Senior Officer, lovely professional university, and attendees. She will be going to discuss about the admission process and other highlights of the university. So thank you so much, ma'am, for joining us today. So with this, we'll begin our today's webinar. So Gurmeen ma'am, over to you, and please take the platform and introduce our guest, our panelist briefly. So over to you, ma'am.
Thank you, Dr. Ruby. Good afternoon, all. It's my pleasure to introduce the two resource persons of the day, Dr. Neeta Raj Sharma, Professor and Senior Dean, School of Bioengineering and Biosciences, along with Dr. Umesh Gautam, Professor and COD, Department of Molecular Biology and Genetic Engineering. So first of all, I would like to introduce about Dr. Neeta Raj Sharma. She completed her PhD from Jivaji University, Gwalior. She has been really instrumental in taking the research-funded projects from national and international agencies. To her credit, she has more than 25 years of experience in research, teaching, administration, and industry. She has also been granted with 25 patents. Thank you, ma'am, for joining us for this webinar. Dr. Umesh Gautam, another resource person of the day. He is a professor and the COD of the Department of Molecular Biology and Genetic Engineering. He is a professional having more than 14 years of experience in teaching and research in the field of plant biotechnology and molecular biology. He has 65 publications in journals indexed in Web of Science corpus. He is having a rich experience in writing research and review manuscripts. Thank you, sir, for joining us for this online webinar. So first of all, I would like Dr. Neeta Raj Sharma to start with the webinar.
Thank you, Ruby. Thank you, Gurmeen, and my colleagues present here. Good afternoon and welcome all the participants in today's webinar. Very interesting title on PCR, a photocopy machine of DNA. I hope all of you are aware about the photocopy machine, that how important it is for any of the offices, any academic institution, or even in the libraries, because by using this machine, one can quickly make several copies of any of the documents within a second. So likewise, PCR, that is polymerase chain reaction, is a photocopy machine of DNA, which is used to generate multiple copies of a particular sequence of a DNA. And it is really very, very helpful to the biologists. And PCR revolutionized DNA research to an extent that the inventor of PCR machine received Nobel Prize. So you can understand that how significant this polymerase chain reaction is in the biology, biotechnology, and several other fields. As we all know, that DNA plays a very pivotal role as genetic material in all our lives, because DNA is unique to every living thing. So biologists often need to make several copies of the DNA, this genetic material, to explore various applications in various areas such as agriculture, molecular biology, genetics, and forensic science is very important. So keeping in view the importance of the DNA detection in current times, as well as the innovation of this PCR long back, but again, there are several variants of this PCR machines available with us. But today, keeping in view to disseminate the knowledge on the PCR machine to our participants, we planned this demonstration webinar for you, wherein you will be learning about the machine, its various components, and how does it work, what are various materials which are required to run the PCR machine, and finally, how you can visualize the various DNA sequence copies. So we have with us for this demonstration Dr. Umesh Gautam from Molecular Biology and Genetic Engineering. He's heading the department. We have our PhD student, Nahid, and we have our lab technician, Mr. Sandeep. So let us have a good learning on PCR machine, and let us learn. We do not know anyone from you people can explore some other application using the PCR machine. So I wish you a great learning of this PCR machine through this webinar. Thank you very much. I would like to invite my team to start the demonstration, or first the presentation telling about few details about the machine, and then let us have the demonstration on PCR machine. I hope you all will enjoy. Thank you. May I invite Dr. Umesh Gautam, please? You are not audible, Dr. Umesh.
No, sir. Still, we are unable to hear you.
Which today we are going to discuss about the PCR, a photocopy machine of DNA. So as we already know about the photocopy machine, that we are taking a paper and we are going to make a multiple copies of the same image. So same here, we are going to do it with this machine, which we call it that thermal cycler. And the technique, their name is PCR, polymerase chain reaction. Why this technique is important? Because this technique has revolutionized that field of molecular biology and biotechnology. This is the man behind that. You can see that on left side, the person who got the Nobel Prize for this invention, this technique that we call it that PCR. This is the year when he got that Nobel Prize. And after inventing this technique, he has totally changed the field of every corner of the science. Is it agriculture? It is medicine. It is a microbial field. Whatsoever it is, the research in human science, so everything has been changed after the invention of this technique. And little bit I'm putting the light on the historical part of the technique. After inventing this technique, Dr. Karamolis, he has published the article on his technique. Now you can understand how difficult, while there is no literature available for the invention, and you are going to write an article. So you student, you can put that your fingers on the Google and you can type the paper of the Karamolis and you can read that paper. So this is available. And after that, there was one more interesting part came into the science, that tag DNA polymerase. This is the enzyme which we are going to use in these PCR reactions. That came into the limelight, and that was the era of the science. In 1989, it was announced that molecule of the year, because it is also part of that. Whenever we go step by step, we'll try to understand. And in 1993, Dr. Karamolis got the Nobel Prize. This is the picture from that when he got the Nobel Prize at that venue. Okay, now why PCR? The question comes in the mind, why we are talking about the PCR? Story has to started with the DNA replication. In DNA replication, that it is going on in vivo conditions in every living organism, that our DNA is going to get replicated. It is going to get synthesized. So in live conditions, for example, me as a human, plants, microorganisms, animals, every living organisms are having cells, and within cells, they are having those nucleus. I'm talking about the eukaryotic cells, and they are having DNA inside. And that DNA, it is going for replication. The idea came from there, that if DNA is going to get replicated or synthesized in vivo conditions, why not in the laboratory? So from that, Karamolis got the idea. I'm going to brief that part, that double stranded DNA we are having, and it is going to get denatured, and newly synthesized strand you can see here. Okay, now a little bit about the principle, because every technique is having their own principle. So it is all about that in vitro. The word speaks about itself. In tube, we are going to run these reactions in tube. And for that, we need a help of one enzyme, which can synthesize that DNA strands. And add on to this, you are going to take a help of primers. We'll talk about these keywords also. Or at the end, we'll get some queries regarding that. Okay, these are the three different steps. If we are talking about this technique, that is divided into three different steps. First is denaturation, second is annealing, and third is extension. Now we'll try to understand what these steps are. How this PCR reaction is divided into three different steps. Whatever which is going on in vivo conditions, same we are going to perform in laboratory, and we'll do it today. The first step, which we are talking about, the denaturation. That is an example actually, where you can see that this is the double-stranded DNA in front of you. And whenever we are providing the temperature, here the temperature range is also given, 94 to 96 degrees Celsius. Almost all the DNA, it is going to get denatured. It means double-stranded DNA, their hydrogen bond in between two, they are going to get denatured, and they are converted into two different helicals. And this is the first step of your PCR reaction. You'll see here. And we have taken one example also. For example, if this is a long DNA, within the DNA, we are having this particular region to get it amplified. That we call it the gene of interest. Now we'll try to understand that how your DNA will be polymerized, and specific target within that genome, how you can get it amplified. So this is about the first step. Double-stranded DNA, you are providing the temperature, and that is going to be happen in that thermal cycler, for which we are going for that live demonstration. This is about the second step, which we are calling it that annealing. In annealing, you can see that there are two different primers. They are going to get annealed. Now the third step coming into that picture, that extension. Extension means now single stranded DNA after denaturation, now it will make the copy of two double-stranded DNA. Now question comes, the tag DNA polymerase enzyme is required to extend this. So how it will come to know that in which direction polymerase will move? Again, I'm going to move. First step, denaturation. Your double-stranded DNA, it is going to get denatured. It is going to open two helicals. Second step, annealing, in which single-stranded primers, they are going to bind with that your gene of interest. Now, the third step coming, that we call it that extension. It happens at the 72 degrees Celsius. Like this. This is the tag DNA polymerase. Now you can understand, once double-stranded DNA, it is going to get denatured. After that, single-stranded primers, they are going to bind it here, and tag DNA polymerase will extend these double-stranded DNA. Like this. But what's happening here? The critical point is that this particular extension is about the copy of DNA, not the copy of our gene of interest. Gene of interest specifies that out of that your complete DNA, you want to amplify a particular stretch. To give an example, you are having your book for that photostat, but out of that book, which is having 20 different pages, but I want to get it photostat page number 4. How I'll get it photostat? Simply, I'll go to the page number 4. I'll flip the pages. Something like it is happening with the DNA. This is the first page of the DNA, which we'll call it first cycle. Just be with me. I'll try to make it clear that point. What's happening with the PCR, which is the most critical point. Done. This is the first page of DNA, first cycle. I have to move up to page number 3 because my photostat copy will be start from page number 4. I do not require page number 1, 2, and 3 to copy. So this is the first cycle of PCR. Done. First cycle contains denaturation, annealing, and extension. It means 3 steps define one cycle of PCR. And after that, it will repeat these cycles again and again. So up to this photograph, we have completed one cycle: denaturation, annealing, and extension, double-stranded DNA. Now we'll move to the cycle 2, like this. Now, how many copies we have formed here? Initially, with one double-stranded DNA, copies of 2 double-stranded, like this. Now we are moving to the next cycle. How many copies we are getting here? 4. Now 2 cycles are complete. Now we'll move to the cycle number 3. Now how many copies we are getting? You will see here, students, there is one difference in cycle number 3. What it is? Just look for the first copy of DNA, this upper part and lower part. This is the copy of DNA only. It means primer is going to bind, it is going to extend. Polymerase doesn't identify where to, it identifies where to start, but where to stop. Don't know. Copy of complete DNA, whatever time we are giving. Now I'll go for the cycle number 2, that copy number 2. This one and this one. Again, it is going to get it extended. But now, can you see that with cycle number 3, this copy, shorter one? This is our gene of interest. So that is my page, which I need to get it photostat. So these are the smaller copies. So what happens if any question raised during PCR, that if we are putting the PCR reaction and from which cycle you are going to get your copy of gene of interest? So the answer will be that we are getting with the cycle 3. Initially with cycle 2, we are only getting the copy of DNA, not the copy of our gene of interest. So it goes with the PCR conditions. Now there are certain interesting things about the PCR. This is the overview, that denaturation, annealing, extension. So now we got our photostat copy. So there are critical points behind that this photostat copy. How many components are there? How we will go and put that PCR reaction? So these are the different components of the PCR. What it is required? First and foremost thing, that you have to isolate a DNA from the organism. This is the DNA. So that DNA, the long DNA, you will buzz the cell, and there are certain protocols, you will isolate the DNA. Second thing, we need a primer pair, forward and reverse, which we have seen for that step number 2, extension. We need nucleotides, which will be added by the polymerase enzyme. As such, polymerase enzyme, and these are the certain tubes, we call it that PCR tubes. These are made up of those polymers. And now this is that PCR machine. In general, we call it that PCR machine, but initially this is, the scientific name is thermal cycler. So we'll put the PCR reaction in this thermal cycler today. So these are the components. Now there are certain interesting facts about this PCR reaction. Now question comes in the mind. We are going to mix so many things. How things will be work in a single tube? So the basic concept behind that, like this. DNTPs, we call it that their full name, deoxynucleotide triphosphatase, which are going to add by the DNA polymerase enzyme. And there is a critical factor in that we need magnesium ion. These are certain components which is required to get your optimum result in machines. What will happen? While you are having magnesium chloride, you are having DNTPs into the reaction. It will make a complex. DNTPs, magnesium, they'll mix with each other and they'll make that complex. And this complex will be the actual substrate. It means we can say if magnesium is not there, substrate will be not available for those PCR reactions. Which is going to identify this man, which we call it that tag DNA polymerase. It is available to identify DNTPs and magnesium ions. Otherwise, it is not possible for tag DNA polymerase to identify the substrate. So this is the actual substrate. By that, this reaction will go on and it will make the copies of the DNA continuously. And one more point we are going to add, and after that we are going for that demonstration of that PCR reaction, how we are going to proceed, and at the end, how we are going to see the result. So this is the optimum concentration. And now one more question comes in the mind. For example, if we are forgot to add the DNTPs, or the DNTPs into the reactions are very less, what will happen? Now we are not live one-to-one. So I'm giving the answer for that. Because in initial reaction, what we are looking for that, if DNTPs and magnesium ions are there, that is going to identify by the tag DNA polymerase. If these are not available or they are in that lesser concentration, that will create a dilemma for that PCR reaction. It is going to get inhibited. Why? What is the reason behind that? What will happen? Because whatever these magnesium ions, they are required for two different things. One, DNTPs is required, and second, if you have read in your biology that enzymes require cofactors. So what is the cofactor for that tag DNA polymerase? Magnesium ions. So what is the food for the thought for PCR? Magnesium ions. So we have to keep that optimum concentration of the magnesium ions in the reactions. Otherwise, we can lose our. So other than that, students, there are so many components are required. Some other time, because we are having that time also that we have to go for that demonstration also. So we can discuss some other time. So that is about that, some key points about that. Now, just ready for that. We are going for that demonstration for the PCR reaction. And after that, we'll run the gel. And at the same time, after running the gel, how we can see the result. So we'll go step by step. So be ready for that. We are going for that demonstration of our PCR. So hope everyone is with me to see that part. Just a second. Hope students, you are with us. So we are having our PhD scholar with us, Mr. Nahid. He's here. He is going to help me to put that PCR reaction. So this is our thermal cycler. You will see.
Sir, I'm so sorry to interrupt you. Can you please stop sharing the screen so that audience can fully visualize
sure, ma'am. Sure.
the demonstration? Yes, sir. Yes, sir. Good to
Fine,
go.
ma'am. It's fine. Okay.
Absolutely.
So this is our PCR machine or thermal cycler. So I'm just going to that. This is the display screen where we are having that program of that. And now I'm going to open this PCR machine. Can you see that these are the blocks where we are going to put our PCR tubes? And additionally with this, you can see that trays are given with that. And these trays, they are going to set into that. And in these trays, we are putting our PCR tubes. And more detail if you want to see about that, this PCR machine. So these are the blocks. These are the blocks. And in these blocks, I'm putting that tray. And this tray, I'm going to put my PCR tubes. And on the other side, you can see that this is a lid. This is a block and this is a lid. While we are putting our PCR tube, we are going to close the lid. And we are going to set our PCR program. So be with us. We are going to prepare. Can you see that screen, students? Can anybody ensure me that screen is visible of the PCR?
Yes, it is visible.
Okay. So now you can see that there is a program here for that PCR. Stepwise, there are different steps which I was talking during my presentation. Stage 1, here written, stage 2 and stage 3. In these stages, they have given certain temperature and time. And here clearly we can see, I can take the help of that my micropipette just to show that this is the temperature, 95 degrees Celsius, and that is the time, 3 minutes. That what we call it initial denaturation. So it means we are giving 3 minutes, total 3 minutes to denature all the DNA available. Now we'll go for a next step. So here we can see that we are again going for a denaturation. I'm putting my pen here that 94 degrees Celsius for 1 minute. This is the step from where our initial cycles are going to get started. So now denaturation, first step, which we have discussed about our PCR for 1 minute. It means we are giving again 1 minute to denature our DNA. Now next step, it will see that is a 55 degrees Celsius for 30 seconds. That is the stage 2. That is the annealing. And last is 72 degrees Celsius for 30 seconds. That is about the extension. Now these 3 cycles will go on. We have set for 35 cycles. So that is about the overview of this PCR machine. Now Nahid will help us to put the PCR reaction. Now, please come with those micropipettes and put that PCR reaction. Now, if you'll see that what Naid is doing, that Naid is having that there is a ice in that, because always we have to get ice and we have to put that PCR reaction into the ice. And these are the different components in these tubes, which is required to put the PCR reaction. So meanwhile, Naid is putting these things. So just stay with us. We are going continuously. Okay, students, now he's going to set up the PCR reaction, and now he's going to add the components one by one. He's adding initially. What knife you are adding? So please speak about that, because it's visible to you, these components. Water you are adding initially?
Yes, sir. Initially, I'm adding water.
Okay. So Naid is adding the water into the PCR tube. He is using the micropipette. How much volume you are taking, Naid? 5 microliter. Okay. Fine. So now he is injecting into that. Done. Second component, Naid, quickly add, because we have to put PCR reaction quickly.
DNTP mix.
Now he is using the DNTP mix. We were talking about the DNTPs, which will make a complex with the magnesium ions. How much DNTPs you are using?
1 microliter.
1 microliter. Okay. Done. Now next component you are going to add into that buffer,
which contains magnesium.
Okay. So now he is using the tag buffer, because tag DNA polymerase enzyme we are using, and for that he is using that tag buffer. Next component, Naid.
Our DNA, which consists of
Okay, DNA template we are using, which we are going to get it amplified with the help of PCR. Good. And now you have to add your forward and reverse primer also. That is also left. Done. Please add. This is a forward primer. Now reverse primer. It's good, Naid. You have already wear the gloves also. That is very much required, because what happens on our hands, we are having dust particles and all, so that it can just contaminate our reaction. So we have to wear the gloves. Yes, please open the lid. Now he's opening the lid and he's putting the PCR reaction. And initially he has to put the tray also, like this. This tray is put into that thermal cycler. Now just put the tube. Done. We are going to close the lid. And this is the PCR program. Naid, please put your finger on start run. Done. Now it is going to run on that machine. Fine. Now we have put the PCR reaction. And now what will be our next step? what we are going to do, because just due to that time, we have to show you these step-by-step process that what will be our end result. So after that, putting this PCR reaction, what we have to do, we have to go for getting this amplified product and to run on the gel matrices, that we call it that agarogel electrophoresis. This is also one of the technique in that we are loading our DNA samples, and these DNA samples are running on those gel trays. We are moving for the next step. This is the bench where your PCR is running. Now next step will go for from PCR to our gel trays. Now, Naik, please show that where are your electrophoresis unit and which is going to start. If Mr. Sandeep can help us about that electrophoresis unit. Yeah, Mr. Sandeep is coming. Yeah. This is the electrophoresis unit. Okay. Now, Naik, please come and show that here you have just loaded your sample, or you are going to load your sample, and that is the power supply. Done. This is the electrophoresis assembly. Done. This is the power supply which we are using to run our DNA sample. And for that, what we are going to do, which type of dye knife we are using to mix the DNA. Now, better if you can show or indicate that your gel matrices and where are the gels.
This is our gel apparatus. So inside this gel apparatus, we'll put our gel. You can see here, it is jelly-like substance made up of agarose. So in this gel, we make wells here. The up you can see there are multiple wells. In these wells, we'll put our PCR mix after the PCR is done.
So now we are already having our PCR mix with us. So maybe it will be the better idea to load the sample directly. Done. So students, what we are going to do, now we are going to load our PCR amplified product, which we have already run. Because due to that, just to save the time, we have done that activity in advance. Because this PCR program, which we have run as a demonstration, it will take 2 hours. So after 2 hours, what we are going to do, we are going to take these amplified product from that tube, and we are mixing one of the dye that we call it that gel loading dye. This gel loading dye, it is mixture of two. You can see that this kind of a blue color dye we are having, which we are going to mix with our PCR amplified product, and we will load those samples in these gel matrices. So Nahid, please do it.
So I'll get 1 micrometer of this dye.
Okay.
So put it on.
Nahid, how much dye you are taking? Just show. Yeah.
1 micrometer.
This is the dot of that dye. Maybe you can take more because it is not visible here. Na? So it's better if you can take more dye. Okay. And now what he is going to do, meanwhile, he is going to mix that PCR amplified product as well as that dye. And this particular sample is going to load in the gel matrices, which he was showing earlier. Now just load the samples. We are having 3 to 4 PCR amplified samples products. So I'll just change the position of camera. So now I just see that your gel tray is visible.
Yes, sir.
Okay. Your wells should be visible. So everybody should see that part, that while you are loading the samples.
So I'm mixing dye with my sample.
And now I just load the dye and your DNA, your amplified PCR amplified product into the gel. And we are having one more sample. Quickly load into that dye. So student can observe that how we are going to check the product on gel matrices. Okay, now that he has already loaded the sample on this gel matrices, that we call it that agarose gel. Now, please switch on the power supply so these samples can run, and we can show that how these samples are running under that voltage. Now, can you tell that what is the signal one will get that your sample is running or not? So it will create a bubbles now around that. Now, please tell the students there are two types of colors and two types of electrodes here. Now, what these are, because we are not able to understand
So
by their color, why these colors and what it is.
DNA is negatively charged, as we know. So it will move from negative side electrode to positive electrode. So our here on
Okay, this is the cathode side. Student, can you see that they have defined with the colors? One is black color, one is red color. So what it is, DNA is a negative charge, we already know. So what we are going to do, we are going to load our sample on the negative side of that. So this is our cathode, this is our anode. So what will happen? Minus will push minus. So it means this cathode will push our DNA to move forward towards the positive side of this red color. So due to that, what will happen? In this gel matrices, you can see that your DNA will run. These blue color dye is visible in the wells. Now it will run. It will take time actually. It will also take time. At least we need 1 hour or 50 minutes to see our result, because dye will separate. At the same time, our PCR amplified product will be resolved on this agarose gel, because this particular technique also required a separate lecture, that agarose gel. And now we'll move where we are going to see our result, where we can see these results. That we call it that next instrument. We are going step by step. First instrument, thermal cycler, where we are putting the PCR reaction. Second step, we are going to check this result of the PCR amplified product. The instrument is gel electrophoresis assembly, and the technique is agarose gel electrophoresis. Now the next step, we are going to move where we are going to check this gel and the result of PCR amplified product. So we'll move for the third instrument, that we call it that this is we are having, because yes, we are rich in that instrument. We are quite lucky that for each and everything we are having instruments are in our laboratory to do the projects of our UG PG students, and as well as to running the laboratories, because science is what about whatever your theory part, you have to prove it by the practicals. And for that, each and everything we are reading about the DNA, RNA, protein, each and everything, and that we have to conduct into the laboratory. So our understanding will be good. So this instrument, students, you can see this, we call it that gel documentation. Okay. So in gel documentation, you can see this is the display part of that. And if I'll open this, you can see that there is a one more part here. If I'm going to open it, so there is one part, we call it that trans illuminator. I'm going to push this translator, if you'll see. Done. Here I'm going to put my gel, but I did not wear the gloves. I cannot pick the gel. Who will pick the gel? Gel will be picked by the student because he has already wear those gloves, which is required to handle these gel matrices. Now, it will be better if you can help to pick the gel from the gel electrophoresis assembly, because students will show you that how gel will be visible on that display, because this is the instrument where you are going to put your gel after running your PCR amplified product, and that result will be visible on this display screen. We'll take 1 more minute to finish this task, because that is the end part of the technique, both results. Okay. So that you can see that how they are going to put that gel matrices into that, like this, they are going to push. Now, this transilluminator, again, every instrument requires that a separate lecture. Now, if you can see the screen here, that the gel matrices will be available. We are going to put the finger, and now you can see that gel. This gel is blank right now because this is the dye which is already available in the wells. Just hold, just hold. Yeah. Okay, that we can show you that our result, which we are already working in the laboratory. Here you can see that these are the wells available, and at the bottom you can see, while you are running the PCR now, these are the primer dimers, one forward, one reverse. If excess primer we are putting into that, that primer is going to be left here. So this is the primer dimer, and there are the DNA bands here, small DNA bands. And we can increase their sharpness by that what? One component, magnesium chloride. So student, this is all about that PCR, which by that we were trying to copy a DNA. So that is our photostat machine for DNA, not for the paper. Done. So thank you very much for giving this chance and to explain our technique, which is the most important technique used in every field of science, Thank you very much. Thanks.
Thank you, sir. Absolutely. Thank you so much for this insightful, informative, and impactful presentation. Your expertise and knowledge in this field is evident from the quality of the discussion we had today and the presentation we have today. And although it was difficult to demonstrate such a useful technique for within 1 hour, but your insights and perspectives provided invaluable information to attendees and helped to deepen their understanding towards this topic. So thank you so much, sir. And so before we commence to other part, we have few queries from the attendees. So, sir, can we take up those queries?
Sure, ma'am.
All right, sir. So, so there is a query from Sai Sahithi Orgwanti. The attendee wants to know that what was the component to for PCR reaction. So I think, sir, this is a technical question.
Okay.
You be able to.
As we have already discussed about that different components of the PCR. So first and foremost thing which is required, that your DNA template. Once we are going to use the word template, it means single stranded DNA. So we need that DNA. We need that primers, forward and reverse. We need one enzyme that we call it the DNA polymerase in general. But in PCR reactions, conventionally, we are using that DNA polymerase. We use another component, that is, we call it the DNTP, which is required to extend in the third step. So these are the overall component, and rest we are going to add the water. And now I want to add on to this query that nowadays we are not separately using DNTPs. We are having a master mix. All the components are there. You have to only add your DNA and your primers.
Thank you so much, sir. So we'll quickly take another query. So that another attendee wants to know that what Hamishka ji, she wants to know that what is the buffer material you have discussed in the presentation.
The basic concept in the practical aspect is that if we are using any enzyme, so those enzymes require the buffers. So tag DNA polymerase, the enzyme we are using, for that, there is a tag buffer which will make that enzyme stable during that reaction. So it can go for its optimum activity.
Thank you so much, sir. So, sir, we'll quickly take another query, last but not the least. Sir, attendee wants to know about the curriculum. Our curriculum of the Department of Specific Programs, they are up to date. They are in accordance with the competitive exams we are having, the syllabus of the competitive exams the students are going to fulfill. So, sir, what's your call on?
Definitely, ma'am. Whatever that query is very relevant, because once we are going for any degree program, so always the question comes in the mind, and that is not about ourselves. This is about the whole university. Each and every department, every program, whatever the requirement of the industry, certain courses are added into that, including those practical aspects. As well as if students wants to go for the higher education, the curriculum is already mapped with those competitive exams. And third part is that if somebody wants to go for the entrepreneurship, so we are having a different unit, but curriculum is up to date according to the demand of industry as well as competitive exams. For example, if somebody is going for that BSc program, so we have mapped our curriculum for the GAT-B exam, IIT JAM. If students are going for their PG program, so we have mapped our curriculum with most of the competitive exams, starting with CSIR NET, ICR NET, ICMR NET, DBT JRF, whatever those competitive exams, our curriculum is totally mapped with it.
Thank you so much, sir. And sir, another query we have, although this was the last and least, but I think we should take this query from Pratik Prohit. He wants to know that the buffer used was acidic or basic.
The buffer is having their ionic strength. All the time, DNA doesn't work in the acidic conditions. So we have to provide the basic conditions for that. But these buffer are having their ionic strength with those positive and negative ions.
Thank you so much, sir. Thank you so much for resolving all the queries of our attendees. And Hamiksha, you want to go through this presentation once again. So I must tell you that this presentation, this video will be uploaded on our website. That is www.lpu.in. There you can go through this video once again, and you can be updated about the other webinars also. So you need to visit at our website, that is www.lpu.in. Moreover, if you have any kind of queries regarding the admission, you want to check your eligibility, are you able to enroll to the program? So you can check through the website. You can check the fees, the fee structure, the various scholarship the university is offering to the students. So you can go through each and everything through that website. That is www.lpu.in. I'm repeating it once again so that you need not forget this ID. And if you have, in case you have any kind of confusion regarding the text, so you can call us. The number is available on the website. You can WhatsApp us. That is also available on the website. Even you can go through the video counseling, or you can drop a mail. So, and the query, whatever the problem you have, that will be resolved at the earliest. So thank you so much, everyone, for joining this webinar. So, sir, with your permission, can we wrap up today's webinar?
Yeah, and ma'am, one more point I want to add here, that whatever we are going for our curriculum and all these things, our School of Bioengineering and Biosciences, they are conducting the training programs and workshops on the regular basis with these recent techniques, where one can get hands-on experience for that. Thank you very much.
Sir, thank you so much. So we have with us our the head of the department of the school, Dr. Neeta Raj, with us. So, ma'am, would you like to give any concluding remarks to our audience, or should we wrap up the this webinar?
Hello, Ruby. First of all, I would like to thank my team that they demonstrated very nicely in a very lucid manner. And I just want to give a message to the students that biotechnology is a very booming area nowadays. And during pandemic also, the area resisted a lot. So keep watching our webinar series, and we are coming very soon with very interesting topic next month. So I wish you good luck. Thank you very much, all of you, for your patience learning.
Thank you so much, ma'am. Thank you so much for your words of wisdom. And so with this, we'll wrap up today's webinar. So my heartfelt gratitude to each and every one of you for attending this webinar on the topic PCR, photocopy machine for DNA. Your presence, your participation and support was instrumental in making this webinar a grand success. And experts, ma'am, sir, you have brought invaluable insights and expertise to the table, and your presentation and discussions were both informative and engaging. And we understand your time is valuable, and we appreciate the effort you took to join us and help to create an environment of collaboration and learning which is useful to the attendees. So thank you so much. And
Ruby, I would like to thank you too. Thank you very much for coordinating very nicely. Thank you very much. And Dr. Gurmeen, thanks to you too.
Thank you, ma'am. Thank you,
participants as well.
A lot from you. And attendees, we look forward to hosting more such informative webinars. So for that, keep a check on our website, that is www.lpu.in. So until next time, I, Ruby, will finally sign off the session. Thank you, everyone.